my classmates and I worked under the guidance of Dr. Bernard Cohen, and we did a systematic review on acne and the cutaneous microbiome. So we sort of broke it down into 3 parts, and we looked at the overall components of the cutaneous microbiome and how it plays a role in acne. And then we looked at existing treatments, so like benzoyl peroxide, isotretinoin, different antibiotics, and how they affect the cutaneous microbiome as a means of modulating for acne treatment. And then also we looked at potential new therapeutics like topical probiotics for acne treatments. Because we had a lot of heterogeneity among the different studies that we selected, we ended up doing a qualitative approach instead of a quantitative approach. And so that makes our study a little bit limited in terms of clinical value. I think we noticed a lot of trends. So obviously Cutibacterium acnes, which is known to contribute to acne, sort of remains like the main player. We found that. But we also found some interesting interactions between Staph aureus, Staph epidermidis, and also changes in alpha and beta diversity, which are indexes of microbial compositions, and how various treatments affect them. It was honestly interesting because some treatments increase diversity, some decrease diversity. So I think that really just leaves room for more like basic science studies on what the actual mechanism is. I definitely think it's a scaffold from which myself and my classmates and anyone who's interested can work from. There's not a lot out there on the cutaneous microbiome. And it's very intricate. So, the different ways that they classify the cutaneous microbiome make it difficult to analyze, but I think future steps involve looking at actual mechanisms for how the modulation occurs and if it's actually contributing to acne or if it's just a byproduct. And also personalized therapies like topical probiotics, and they found some interesting like plant strains and different topical therapeutics that function as a means of modulating the cutaneous microbiome for acne treatment. Just the beginning of our research, we found that there's something called alpha diversity, which is the difference within a sample of like the bacterial communities, and that tends to be increased, meaning more diverse, with healthier skin, and decreased in acne lesions. What we found to be surprising was a lot of the studies we found the opposite finding, and also several treatments, so benzoyl peroxide, for example, all of the studies that we found were effective at clinically improving acne, but they decreased the alpha diversity, whereas other treatments would increase the alpha diversity. So I think that was the most interesting, was those diversity index changes, and I think it's worth future studies.